Myself Minoti Upasani, and the topic for my paper is Global Proteomic Based Identification and Validation of Thymosin beta 4 X linked as a prognostic marker for head and neck squamous cell carcinoma. So the author who have published this is utilising. He studied from Tabai Municipal Wanfang Hospital. He is currently working there. He has studied his master's in biomedical informatics in 2013 and is currently a PhD candidate working in a genome research center in the same institute. He have contributed for biological sciences like in genomics, biochemistry, molecular biology, and the highlight of his research areas are bioinformatics, and there are some genetics and protein interactions. So this paper have completely focused on a particular type of cancer, that is head and neck squamous cell carcinoma. When we say about cancer, we understand that it is something that is not regulated, and because of that there is cell divisions, and there is something called as tumor which gets formed. It is, if it is in a localized region, then it's not a problem. But when it is a metastatic, it can spread across the body into different parts. So that's why it's important to understand. When we say about head and neck squamous cell carcinoma or carcinomas in general, they are one of the types which are affecting the epithelial cells, and specifically squamous cell carcinoma are affecting squamous cells. So under this category, this is one of the cancer, HNSCC. Then this is a sixth most common cancer which we can find worldwide. And what can we observe in these patients is like abnormal patches, unusual bleeding, pain in mouth, sinus, enlarged lymph nodes are some of the things which we can observe. So these are the different areas where a person can get affected. So thymosin beta 4X linked, this is the biomarker that is being found in this paper. This biomarker is being found for other tumors or other cancers conditions as well. So it must have been considered because of that. It is a actin binding protein. It is responsible for different sort of functions like cell migration, adhesion, wound healing, stem cell formation. These are different functions. Basically, it is regulating something called as F-actin and G-actin, and when there is a problem in the regulation, it can lead to tumor formations. So that's why its expression is very necessary to be monitored. So if it is having elevated levels, then we can see these elevated levels mostly in case of the malignant tumor tissues. So this head and neck squamous cell carcinoma is mostly seen in case of oral cavity, oropharynx, hypopharynx. And what can be the predisposing factors like viral infections, alcohol, carcinogens, physical irritations, or host susceptibility? Then treatments currently are like surgery or radio therapy. These can be coupled with the chemotherapy, but these treatments are not for a long-sighted vision. So when we say something we need to treat is by some permanent solution or some more treatments which can be effective against it. This can be done by using biomarkers, and how we can identify it by proteomic analysis. When we do proteomic analysis by using different methods like MAR-DIMS or mass spectrometric analysis, we can find these biomarkers and we can act against them, and we can understand the cancer maybe in the earlier stages. So this TMSB4X expression levels in normal cells are comparatively lower, but when we compare it to HNSCC samples, it can be found higher for those who are in late stages. So what all methodologies they had performed is, first they took cell lines and cell cultures, what conditions they maintained. So they have taken 7 HNSCC cell lines and they were made in DMEM, and normal human oral keratinocytes and oral mucosa fibroblasts were kept as controls. Then patients, 86 patients were taken, and 35 among them were normal patients, normal people. Then protein lysate or SDS page was performed, where cells were lysed, centrifuged, and SDS page was further performed. Then LCMS analysis was done, where with the help of LTQ tandem mass spectrometer, which is based on ions and charges, their peaks would be observed. These peaks, whichever the peaks were obtained, which was prominent, those peaks were considered. So whatever data was acquired, that data was basically compared with IPI, which is a standard set of thing with which we can compare and identify which peaks to be considered. So tissue samples were taken for MALDI. For fresh frozen tissues were cut thaw and they were fixed. Then MALDI-IMS imaging was done. Initially, it was calibrated by insulin or cytochrome C, and then average mass spectrum of each of these were taken, and statistical data were also, statistical analysis was also applied. And there is something they performed is immunohistochemistry. Immunohistochemistry is one of the thing which is commonly performed in case of cancer cells or cancer biology studies, where the histological samples, the way the histological samples are stained with H and E staining. Similarly, what happens is the antibodies which are there, they are present, and the antigens in case of tumor cells, whatever the reaction happens, they are then further stained and then they are visualized with the help of immunohistochemistry. So it can be observed that all of the 4 stages were taken for this, and the tissues were formalin and paraffin fixed, and formalin fixed and paraffin embedded, and then were further analyzed. For validating this particular study, real-time PCR analysis was done, where total RNA which was there, that was extracted, reverse transcriptase was performed, and then the mRNA level for this TMSB4X was checked with the help of the cyberdyes. Then cell proliferation assay and cell mobility assay. Cell proliferation assay will give us how much count the cells are having, like after a couple of days, how much count is there. Or cell mobility and invasion assay will tell that how metastasis is occurring. Then orthotrophic xenograft mouse experiments, where basically the knockout mutations were carried out. So this SAS cells, they were transfected with a luciferase plasmid, and these cells were inoculated into the mice. Now, what was this particularly? They knocked out the gene for TMSB4X, and then they checked what can happen. So new techniques or instrumentations which have been seen is LTQ tandem mass spectrometer, the instrumentation which is used mostly over here. And invasion assay is a technique which was new to me. Invasion assay is a particular type of technique where on the upper side, the cancer cells are seeded, and below that, any chemoattractant is being added. And because of that, if they invade the surrounding, that can be observed, and they can further be analyzed by some fluorescent dyes or something, which is basically done to check whether there is any metastasis occurring, whether how it is affecting its surrounding, or basically if there are any drugs that can be used against it, or how invasion can be stopped, all of these things. So what was observed is, first initially they compared all the data sets that they got and they identified it, and there were about 11 samples that were selected. So 2 normal mucosal specimens and 9 HNSCC specimens were taken. To differentiate between them, they did H&E staining for cancerous and non-cancerous. And this is what they observed in that all the stages, there is more prominent early fluorescent dyes or more prominent seen in the stage 3 and stage 4. When we say these two normal cells, that is OMF and NaOH, NHOK, sorry. Basically, these are the normal ones, and the cancer cell lines like TW1.5 and Cal27 were taken. So, what can be observed here? These are the intensities. These are the peaks of interest. We can observe that these two are the normal cells. So there is not a significant peak that can be observed. But when we observe in these two cell lines, which are cancer cell lines, we can definitely see that there is a good amount of peak generated. So there was high intensity as we observed here. This is Cal 27. There was a great amount of peak that is observed. And in normal cell, it was less. And as we saw that there is high intensity in case of stage 3 and stage 4. Peaks were observed at 4968.1 M by Z, which is a peak, which is basically the value for TMSB4X. Now, further they validated by RT-PCR and real-time PCR, where they check for the expression that how tumor cells and normal cells are expressing this particular thing. Now, what we can observe is on the y-axis, this is TMSB4X levels, and the x-axis normal and tumor cells. So whatever we observe is the values from the normal to the tumor are constantly increasing. In all of the cases, it's the same. It is constantly increasing. So we can say that expression as compared to normal to the tumor, the expression is more in case of tumor cells for TMSB4X. So even the p-values were significant here. Then in immunohistochemistry staining as well, TMSB4 had elevated levels, where if we can observe it, immunohistochemistry, this is comparatively lighter. So, and this is the tumor cells which are comparatively darker. So stain has been weakened up, and because of which the tumor cells are appearing very darker because this particular TMSB4 is present in it. And TMSB4X is expressed in both of the RNA and protein levels. And these patients were later categorized with the help of whatever IHC scores they got. Now, mRNA expressions for this was done in lentivirus, where lentivirus shRNA, it was seemed successful, where they suppressed this TMSB4X in SAS cells. What they mean here is that we can observe here on the y-axis there is TMSB4 levels. On the x-axis we have different cell lines. So in which this SAS is having a more number of expression, TMSB4 expressions. So when we observe here, that time after taking this SAS, this is the normal. This is the normal one where knockout is not being done. And these are the two subtypes where the knockout is being done in the mice. So we can observe that there is an asterisk mark, we can observe that there is a significant difference between both of them, which can definitely mean that there is less expression of TMSB4 when the knockout is being performed. Even when we compare to the duration, here in a number of cells on the y-axis and on the days on x-axis, we can see that where the knockout is not being done, after some days, there is more number of cells if we compare to the one which was having knockout. So the cell proliferation which we say over the time is comparatively less over here. And then the invasion assays which were performed, in that as well, we can say that this is the normal one, and these are two, the one where knockout was performed. We can see that there are very less number of cells present, and here as well we can see there is a significant difference. So in the cell functions also we saw the, like this, observed the cell proliferation reduction, and because there is cell proliferation reduction, which definitely means that TMSB4 was responsible for enhancing this particular cancer for metastasis, and it was observed that there is reduction in invasion ability for SAS cells, and when it was observed for cervical lymph node metastasis, it was reduced in that case as well. How did they demonstrate this? Basically, how did they observe it is by bioluminescence. So, in mice, wherever the knockout was performed, uh, what they did is by bioluminescence they observe this is the minimum and this is the maximum level of it, how the metastasis is taking place, and over the period of weeks they observe. Here, when the knockout is not performed, we can see that the maximum intensity and it is spread in the area where in the neck area it is being spread. Where here we can see it is pretty much not, compared to this, it is not that much spread and is having a less type of intensity. So that was observed by them. In conclusion, we can say that TMSB4 expressions which are upregulated in case of HNSCC patients or HNSCC cells. And this expression can be detected by or validated by IHC staining and RT-PCR. Whenever there is knockout done of this particular gene, their cell proliferation is also being reduced, and cell mobility and metastasis is also regulated by them. So if therapeutic, it can be a therapeutic candidate where we can focus onto inhibiting this particular factor. The key findings of this paper were that the intensity of Cal27, that is one of the cell line on stage 3 and stage 4, was comparatively high for TMSB4 expression. And there was elevated levels of this in tumor cells. After knocking out, there was reduction in cell proliferation. These were some of the key findings. What I liked about this paper and what I understood is that I was unaware about what HNSCC carcinoma is or what can be the factors leading to it. So it gave a clear idea for me. Then biomarkers like TMSB4X, I was knowing about biomarkers, but how they act, how the action is, how do we analyze it, and how expression of this can affect was understood well. And after knockout mutation, also how biomarker expression can be affected, that was understood as well. What can be some future prospects are, there are different like administration, how drug administration can be done against these TMSB4 biomarker, or in clinical trials, how can we check for this particular biomarker, or how the overexpression of this particular biomarker be controlled. That are some of the things. And thank you so much.